Thank you for the introduction. And my name is Dongju Lim, and I'm currently a PhD student in Department of Mathematical Science in KAIST, and a member of Biomedical Mathematics groups in IPS. And today, I'd like to talk about the noise locus perfect adaptation in biomolecular systems. Before going on to the main topic, let's start with really simple example. If we do the exercise, then our heart rate will goes up like this red line, but we don't want our heart rate to stay at this high level. We want it to come back to the low level, original level, so that we can live in a normal life. Like similarly, if we eat chocolate, then our glucose level will goes up. But similarly, we want it back to the original level. So like these two examples, the systems maintain its stable output even after some kind of perturbations is called robust perfect adaptation, RPA in short. And this RPA property has been a desired property for the biological systems. So to achieve the RPA for biological systems, many biological models has been proposed. And one of the promising biological model is antithetic integral feedback loop. In this antithetic integral feedback loop, if this XL is the output of this system, then this XL produces the G2, and this G2 sequestered with G1, and this G1 produces the X1, which is the input of the system. Then, if once this XL, our output increases, then it increases the G2, so it decreases the G1, so it decreases our input, so it can finally decreases the output. So from this like compensation, we can control the output of the system using this antithetic integral feedback loop. More mathematically, we can understand this compensation by moment equation. So this moment equation, from this moment equation, if this system goes to the stationary distribution, then these two equations become zero. So by subtracting these two equations, we can know that this mean of this output become this mu over theta. An important thing here is that this mu and theta is the parameters of the controller. More precisely, this is the parameter of the antithetic integral feedback loop. So it means that by adjusting this parameter of the controller, we can determine our desired level of output mean. So in other words, we can control the output mean. So even if the original network shows a increase in the output level, like this gray line here, if we attach this antithetic integral feedback loop, then like this blue line here, we can achieve the stable output even after the perturbation. So we will call this as a mean controller, as it can control the mean. But one like major limitation of this mean controller is that it does not control the noise of the output level. So like this figure, after the perturbation, the output noise increases. And more importantly, if we compare the noise level with the original level, original network, that it amplifies the noise level. And this noise amplification actually hinders the like precise control of the output. So for example, let's say that we want to control the protein level at 10. Then by using this mean controller, we can control the level of this protein at 10 in like population average level. However, if we see the protein level in each of the cells, then because of this amplified noise, it shows large deviation from the set point, our desired set point, which is 10. Therefore, it shows that if we want to control this protein level for each cell, then we should also control the noise, not only for the mean. So to control both mean and noise, we made other additional controllers, which we will call noise controller, NC in short. So this noise controller has similar structure with mean controller, but it differs with that it degrades the input species of the system, and it senses the output from the dimerization of the output. And if we form the noise controller like this, then the good thing is that actually we can derive the formula of the bound vector of x2 at the stationary distribution with this whole system. And important thing here is that again, this bound vector formula of the x2 is only determined by the parameters of controller. So this mu1 and zeta1 is the parameter of MC, and mu2 and zeta2 is parameter of the NC. So it means that by adjusting this parameter of controller, we can actually control the output of bound vector as well. Therefore, by combining this noise controller, we can achieve stable output mean, and also we can achieve stable output noise even after the perturbation by using this noise controller. So far, from the original network, if we attach the mean control, then it amplifies the noise, but by combining the NC, we recover this noise at the original level. Then the natural next question will be can we further reduce this noise by using an NC? And we thought that it is possible because if we see the bound vector formula here, then by reducing this parameter of the NC, then we can further reduce the bound vector of x2. Therefore, we set the different level of target bound vectors by adjusting this mu2, and we actually simulated if we can achieve that level of bound vector. When our target bound vector is above 1, then we actually achieve our desired level of target bound vector like this. However, if the target bound vector is below 1, then we could not achieve our desired target bound vector. And as you can see in this really dark green line, bound vector does not go below the 1. Well, we guess that this is because many natural biological systems show the bound vector greater than 1, as shown in these two figures. So it means that maybe our NC can reduce the noise level as much as desired, but up to the natural limit of bound vector of 1. So, and we further checked if we can achieve this kind of noise reduction for different primary conditions. So to do that, we adjusted the target mean and target bound vector value by adjusting the parameter values of MC and NC. And what we did is we simulated the bound vector and we compared it with our target bound vector. and we draw a heat map for the ratio of this simulated bound vector and the target bound vector. And in this heat map, this white area means that simulated bound vector is same with target bound vector. So it means that we successfully achieved our desired level of bound vector. So what can we know from this heat map is that if the target bound vector is above 1, then we could achieve our desired level of bound vector. But if the target bound vector is below 1, then we cannot achieve our desired target bound vector, but it stops at the bound vector of 1. So we also checked if we can achieve this noise reduction up to the bound vector of 1, even with the other biological networks. So previous simulations were done by this simple biological network. But if we replace this original network with this biomolecular network, then we still see the noise reduction up to the bound vector of 1. You can see this white area above the target bound vector of 1. And for even more complex networks like this, we got the similar result that we can reduce the noise up to the bound vector of 1. So it clearly shows that NC can reduce the output noise up to the bound factor of 1. But important thing here is that our the main goal is achieving the output control in a single cell level. And even with this low bound factor, there is some possibility that we could not achieve the single cell level control. For example, if the output distribution looks like this bimodal distribution, then the output level will goes up and down like this figure. Then even with the low bound vector value, because of this jumping behavior, it will show large deviation from our desired set point. Therefore, we further checked if our NC can prevent this bimodality of the output distribution. So to do that, we utilize the toggle switch system. And this toggle switch system has 2 production modes. One is the best production mode, where the repressor B is not bound to the gene A, so gene A can fastly produce the A, our output protein A. And the slow production mode is when this repressor B bound to the gene A, so it slowly produces the protein A. So because of these 2 production modes, this output level goes up and down depending on this transition of these 2 modes. And eventually it leads to some bimodal distribution or the output distribution. When we use only MC to prevent this bimodality and concentrate the distribution to our set point, then still, as you can see in these blue figures, it still shows the bimodal behavior. However, if we use both MC and NC, as you can see in this figure, it shows the unimodal behavior so that it does not show much deviation from our desired set point, which is the dotted line. So this example shows that our NC can prevent the bimodality of the output distribution, and it means that it shows that NC can actually control the output in single cell level. Then the next question is, how can we use this ability of NC in the real biological systems? To illustrate the potential of NC, we adopted the DNA repair system. So in DNA repair system, this AD protein is the crucial species for DNA repair. And once the DNA damage comes into this cell, then this ADA starts to produce its own mRNA. Therefore, this reaction forms a positive feedback loop between ADA and its mRNA. So because of this positive feedback loop, it produces more and more ADA protein. So it helps the DNA repair. Important thing here is that this positive feedback loop will work only when this number of ADA protein is non-zero. So if the initial distribution of the ADA proteins is look like this, then after 1 hour of the DNA damage, the distribution of ADA protein will be changed like this distribution. So for this red area, for the cells without any ADA protein, because they cannot operate this positive feedback loop, so it cannot generate any ADA protein, so it failed to repair the DNA. However, for the cells with positive number of ADA proteins, as they can operate this positive feedback loop, they can produce more and more ADA proteins. I mean, they can produce enough ADA proteins, so they succeed in repair the DNA. Therefore, what we wanted to do is we wanted to reduce the ratio of this failure of the DNA repair. So to do this, we have to decrease the probability of having no ADA protein. So to do this, we utilize MC and NC. Specifically, we set it the output mean and output bound factor so that we can make somewhat right skewed distribution like this. So by using MC and NC, we could shift the initial distribution of ADA protein from this to this right skewed distribution. Therefore, we could reduce the probability of having 0 ADA so that after 1 hour, if we check the ratio of failure of DNA repair, we could check that this portion of the failure in DNA repair reduced from 20% to 7%. So in summary, this all examples shows that our NC is a powerful tool for controlling noise, like this figure. And one intuition between why this NC work well is something like this. So if we only use this main controller, it is same with we're using only one pulley to pull up or pull down the like one box, X2 box. Because if we only use this main controller, then there is only one reaction which affects this original network. So it is same with just using one pulley. However, so as you can see in this figure, if you use only one pulley, it is somewhat unstable. However, if we also use this noise controller, then it is same in using an additional pulley, which pull down this X2 box. Actually, because this NC degrades this X1, it is same as adding the additional pulley, which pulls down this X2. Compared to this situation looks intuitively, this situation is more stable and it is more easier to control this level of X2. So we think this is the kind of one intuition why we should use NC to control the noise of this X2. But one limitation of this study is that we know that we cannot reduce our final factor below the final factor of 1. And more important thing is that if we set our target final factor below 1, like this, then actually there is some species diverges, so they accumulate forever. So it may indicate that this combined system, this combined network, is not ergodic if we set the target final factor below 1. So even though we could not theoretically prove this fact, but we think that we need further theoretical analysis to strengthen the understanding of NC regarding this statement. And another limitation of this study is the biological realization of this NC. So this figure is the biological realization of the MC, which is made in previous study. In this biological circuit, our output is X2, and this X2 acts as an transcription factor of anti-sigma factor here. And this anti-sigma factor sequesters with this sigma factor, which is produced with a constant rate. And this sigma factor acts as a transcription factor of X1, which is an input species of our network. Because MC and NC shares a similar structure, we can make the biological circuit similarly, like this. First, this dimer of X2 acts as a transcription factor of this anti-sigma factor. And this anti-sigma factor should catalyze the degradation of this X1. But the one challenging thing here is that this anti-sigma factor has 2 roles, which is it has to sequester with this sigma factor, and it also has to catalyze this degradation of X1. So finding this some molecule or protein which can do these 2 roles simultaneously is really challenging. So if we can find that kind of molecule, then I think we can realize this NC. And if we can biologically do experiment with that biological circuit of NC, then it will further demonstrate the power of the noise controller. So, and I want to thank our collaborators, Dr. Yoon-Min Song and Professor Jae-kyung Kim in our group, and Mr. Seok-Han Moon and Professor Jin-Soo Kim from Postec. Thank you for listening.
Thank you. Thank you for